So first up today, we have Amber Johnston. She received her bachelor's in animal science from Michigan State University in 2019. She's been working at the Michigan State University Aquatic Animal Health Lab since 2017. Let's see. And was admitted to the graduate school in 2019. She's interested in understanding both the role of infectious diseases play in fish populations and how they influence conservation and management decisions. So with that, Amber, you can go ahead and take it away.
All right. So I would first like to thank the organizers for putting this opportunity to get together, because as a first-year master's student, it was pretty disappointing to have all the meetings I was planning on going to this year canceled. So I greatly appreciate having this opportunity to present what I've been doing for the last year. I also want to acknowledge my co-authors for their contribution on this research they come from the Michigan State University Aquatic Animal Health Lab, UC Davis, Michigan DNR, and the MSU Molecular Ecology Lab. So before I get into things, I'd just like to briefly introduce myself. I'm from a small town in eastern Michigan. I grew up on the Great Lakes, so it only makes sense that I'm here doing what I'm doing now. I joined the Aquatic Animal Health Lab as an undergrad in 2017, and during my time there, I'd really fallen in love with what I was doing, and I actually took on an independent research project working with Dr. Locke and Chris Knupp, doing some work with flavobacteria. And like I said before, I really enjoyed what I was doing, so it all kind of culminated into me being admitted as a graduate student in the lab in 2019, and that's where I am today. So the U.S. Fish and Wildlife Service is funding my thesis research that will aim to uncover the risk emerging and endemic fish pathogens have to Great Lakes lake sturgeon populations. To kick things off, lake sturgeon are an ecologically and culturally important fish. They have a series of symbolic meanings nationwide, and they are, as I'd like to describe, the charismatic megafauna of the Great Lakes. To make them even more unique, they're also the largest and longest living fish in the Great Lakes Basin, with the lifespan reaching up to 150 years. And unfortunately, for reasons I'll discuss in the next slide, their populations are currently less than 1% of their historical numbers, warranting protections in most states or provinces in which their indigenous populations reside. The lake sturgeon in the Great Lakes face a multitude of natural and anthropogenic challenges. Their unique life history traits, such as sporadic spawning, delayed maturation, and low recruitment, put them at a heightened susceptibility to anthropogenic factors, such as dam construction, habitat degradation, and overfishing. And for these reasons, among others, there have been substantial resources allocated to raising Great Lakes lake sturgeon, whether it be in streamside rearing facilities shown here or brick and mortar hatcheries. Alarmingly, it's not uncommon to have occasional sizable mortality events at some facilities raising these sturgeon. Mortalities have been observed at state, federal, and tribal facilities all around the basin, with extreme cases reaching near 100% mortality. At a glance, most everyone who is rearing lake sturgeon in the Great Lakes Basin is grappling with some degree of mortality at some point or another. And unfortunately, using routinely employed diagnostic methods, the causes are often undetermined. And this is a huge problem because not only do these mortality events negatively affect the conservation efforts, they also have significant negative economic effects. So while this is happening, farming of other sturgeon species for human consumption and conservation has increased in other regions of North America, specifically out west and in Canada. Concurrent with those efforts involving other sturgeon species, we've seen an increase in reports of sturgeon pathogenic microbes that lead to substantial losses in those regions. And alarmingly, although we don't have rigorous surveillance efforts in place, these reports have suggested an encroachment from the north and the west towards the Great Lakes Basin. And to make all of this even more complicated, because the diagnosis of these pathogens often requires specialized diagnostic techniques, like I mentioned before, we haven't even extensively looked for these pathogens within the Great Lakes. And that begs the questions, are they there? Are they not? Nobody can really answer that right now. And that's where this study hopes to answer some questions. So our overarching hypothesis approaching this research is that infectious diseases negatively affect the survival, growth, and recruitment of Great Lakes lake sturgeon populations. Supporting that, we posit that by highlighting those pathogens that pose the greatest risk to lake sturgeon, including those not yet present in the Great Lakes, effective prevention and control strategies can be designed. This hypothesis is founded off of a few key points. The first is that we must uncover the role infectious diseases have on lake sturgeon populations, because as of now, we have a very minimal understanding of that. And even more problematic is that something is going on in these rearing facilities from time to time, and we don't know what that is. And unfortunately, because we don't have a very comprehensive understanding of these sturgeon pathogenic microbes, especially in the context of the Great Lakes, we don't even entirely know if our current prevention and control methods are working. So for the next few slides, I'm going to go over the research that will be making up my master's thesis. I'm going to go through what has been done, what I plan to do, and then I'm going to take you through some interesting findings to date. So to begin addressing our hypothesis, we developed 3 key objectives. The first is to determine the prevalence of pathogens in Great Lakes lake sturgeon. Next, we'll elucidate the susceptibility of juvenile lake sturgeon to endemic and emerging fish pathogens. And third, we'll assess the susceptibility of sturgeon pathogenic microbes to routinely used hatchery disinfectants. Starting with Objective 1, adult lake sturgeon were sampled at 3 different locations within the Great Lakes Basin, encompassing the Huron, Michigan, and Lake Erie watersheds. And since we know many pathogens affecting other sturgeon species require specialized diagnostics, we decided to cast a wide net that will ideally identify bacterial, fungal, and viral pathogens alike. We also sought out to deploy methods to link the detection of a pathogen with the lesion. So based on what we know of other sturgeon pathogenic microbes and their affinity for the skin, we sampled fin and gill tissues. We also sampled gametes to further investigate the possibility of vertical transmission. Next, we collected and analyzed blood, as this could help us determine if the sturgeon was grappling with a systemic infection. And lastly, we sampled any gross external lesions that we came across. We came at these samples from multiple angles. Once tissues were collected, we sought out to employ a series of non-routine diagnostic methods to uncover the presence of any sturgeon pathogenic microbes. Next, we sampled fish from 4 different streamside rearing facilities that were largely populated by fertilizing eggs that were collected from the sampled adult populations. And this approach will let us determine the infection status of the juveniles and how exactly that relates to the infection status of the adults. Fish were collected once 5 to 6 weeks post-hatch, and again, just prior to stocking. After euthanasia with MS222, internal organs were collected for virology and bacteriology, and fin, gill, and/or skin tissue was collected for virology, histopathology, and molecular work. So one of the next phases of this study will be to elucidate the susceptibility of juvenile lake sturgeon to endemic and emerging fish pathogens. As I alluded to earlier, there are a number of very devastating sturgeon pathogens elsewhere in North America. And among some of the most damaging that we know of are acid-centered herpes virus 1, acid-centered herpes virus 2, white sturgeon iridovirus, and frog virus 3. And by damaging, I mean that, for example, white sturgeon iridovirus has led to almost 95% mortality in affected lots. Another concern is the fact that some of these acid-centered herpes viruses have been found in gametes and have caused consequential mortality in the resulting juveniles. So I'm telling you that these pathogens are spreading, and what we don't know is what would the effect of these pathogens coming into the Great Lakes be? How would any of these pathogens impact our already at-risk and highly treasured lake sturgeon populations? We don't know that. So to begin addressing the unknowns, we'll be collaborating with Dr. Esteban Soto's laboratory at UC Davis on several aspects of this study, including experimental challenges. These experimental challenges will utilize the 4 pathogens I mentioned before, the 2 herpes viruses, white sturgeon iridovirus, and frog virus 3. Next, in our lab, based on previous surveillance mentioned before, we're going to identify 4 pathogens that we suspect are negatively impacting Great Lakes lake sturgeon, and we hope to uncover how the pathogen behaves in an infected host. Another future phase of this study will be to assess the susceptibility of sturgeon pathogenic microbes to routinely used hatchery disinfectants. This final and critical experiment will arm field and hatchery workers handling Great Lakes lake sturgeon with the proper tools needed to prevent or control the spread of infectious diseases. This third objective is basically to determine how we can prevent transmission of some of these pathogens during the process of hatchery rearing. For example, when eggs are being collected from wild lake sturgeon, how can we ensure that should those sturgeon be infected with something, the pathogens coming with those gametes are checked at the door? And this is all exceptionally important because in the Great Lakes Basin, all lake sturgeon conservation efforts are based exclusively off of gametes collected from wild adults. So we need to make sure that we're doing the best we can to ensure whatever comes in with those gametes doesn't affect any of our rearing efforts. Going off of that as well, during our adult handling events, how can we be sure that we aren't unintentionally transmitting any pathogens from fish to fish via handling or the repeated use of equipment? We don't have answers to those questions, and we need to figure them out. So next, I'm going to bring you through some interesting findings to date. First, year one of adult sampling has been completed, and actually the day after I finished my undergraduate degree, literally the day I walked across my stage to get my degree, I set out to begin sampling for my graduate studies. And while it felt like a lot then, because of that, I was effectively able to collect blood, fin biopsies, gill biopsies, and lesion tissues from over 230 adult lake sturgeon. And while I have a lot of analysis ongoing and have recovered a lot of microbes, I have one particularly significant finding I'd like to discuss in the next few slides. In the process of sampling these fish during year one, we observed these circular, raised, semi-translucent areas of apparent cutaneous thickening. The lesions varied in location, sometimes on the fin and sometimes on the body, but they also varied in intensity. Sometimes they were more localized or focal in nature. In other cases, they were multifocal. And in some fish, the intensity of these lesions was far greater and seemed to be in a coalescing nature. In terms of how frequently we saw these lesions, they were most prevalent in the Black River, but were also observed at a prevalence of about 1% in the St. Clair River. However, none of these lesions were observed on fish handled at the Peshtigo River. We also wanted to look at these lesions histologically, but before we get into that, I wanted to show you a skin section collected from a grossly normal area, in which you can see an epidermal layer of normal thickness and the underlying dermis. When we compared that to an affected area, the first thing we noticed right off the bat was how thickened the epidermis was. In other words, there was significant epidermal hyperplasia. We also noticed the presence of an inflammatory infiltrate within the epidermis, but it was more pronounced in the underlying dermis, at least in the subset of tissues that we had looked at. In other areas of these lesions, we also noted swollen degenerate epithelial cells, shown here with the red arrows. And lastly, we also noted prominent intercellular edema, which some of you may have also appreciated in the previous slide, but it's clearly indicated here with the red arrows. The gross appearance of these lesions, in conjunction with our histological findings, very much resembled what's been documented in other asp-censored herpes viruses and other sturgeon species. But just to remind you, there are no published reports of asp-censored herpes viruses in the Great Lakes Basin. And for that reason, we began pursuing specialized diagnostic methods. Now that we had a lead, we could start working towards finding an assay that worked to figure out what we were dealing with. First, nucleic acid was extracted from suspect lesion samples. Following that, we employed a PCR assay that utilized a degenerate primer set targeting the DNA polymerase gene developed by Dr. Larry Hansen. We decided to use this assay due to its ability to detect a wide array of viruses affecting fish, including asp-censored herpes viruses and others. The resulting amplicon was then sequenced, and DNA was phylogenetically analyzed. To quickly orient you, we had positive amplification in our two controls, asp-censored herpes virus 1 and asp-censored herpes virus 2, and we also had amplification in the majority of the lesion tissues that I had assayed for. We had 4 samples from Black River that came back as positive, and 1 sample from the St. Clair River. So what you're looking at here is a dendrogram showing the phylogenetic relationships between a multitude of reference strains, including as-spencered herpes virus 1 and as-spencered herpes virus 2, and my lesions sampled way over to the right. And here's the exciting part. All of my sequences formed a robustly supported clade together. And although most similar to as-spencered herpes virus 1, the analysis clearly showed that these are quite distinct from currently described as-spencered herpes viruses. And this is all highly suggesting that I've detected a never-before-described as-spencered herpes virus. And this is important for a few reasons. First, there are currently no published reports of as-spencered herpes viruses in the Great Lakes, making this a significant finding, particularly because as-spencered herpes virus 1 and 2 are listed as emergency pathogens according to the Great Lakes Fish Health Committee model program. Second, I told you how problematic the other as-spencered herpes viruses are in other regions of North America, but what effects this virus has on Great Lakes lake sturgeon is almost completely unknown and needs to be addressed. And lastly, other as-spencered herpes viruses can be transmitted from parents to offspring. So is this also a risk to the offspring coming from these adults? We don't know that, and we need to figure it out. So that was a very exciting finding, and as you could probably imagine, I was extremely excited to get back out into the field this year. However, the outlook of that happening amidst a global pandemic was fairly bleak. It took a lot of work and effort from a crew of highly determined people, but nonetheless, I was actually able to travel to one of my field sites on about 72 hours notice. I was able to travel to the Black River, where I'd sampled the most fish in the year prior, and salvage some of my 2020 field season. And I'm going to be completely honest, hiking a river, all the while decked out in proper PPE, was not the most enjoyable thing in the world. So moving forward to what I saw while I was in the field, it was largely similar to the year prior. I saw multiple fish exhibiting these as-spencered herpes virus-linked lesions, and while the Black River crew and I did handle a significantly smaller number of fish this year, we observed these plaque lesions at a prevalence of approximately 14%. Analysis of these samples is still underway, being that I returned from my field site about a week ago today, so we're still working through all of these. However, I do have a few conclusions thus far. The first is the detection of a potentially novel as-spencered herpes virus in the Great Lakes Basin. And second, I'm still very early on in this study, and while I focus in on this as-spencered herpes virus today, I have hundreds of bacterial and fungal isolates that I have yet to analyze, including some that are flavobacteria. So looking at the anticipated outcomes of this study, the first is to identify sources of unexplained mortalities. We still have something going on in these rearing facilities from time to time, and we still don't have a good idea of what that could be. But what we do have now is a series of many microbes recovered from these adult and juvenile lake sturgeon that could potentially be culprits, and we hope to answer those questions. Next, we'll elucidate the susceptibility of lake sturgeon to emerging and endemic pathogens, and this is now exceptionally important with this recent discovery of a potentially novel as-spencered herpes virus. We have absolutely no idea what this virus could be doing to our juvenile lake sturgeon or our adults, and we need to figure that out. And lastly, we hope to devise disease prevention and control methods to implement in both hatchery and field conditions. And this is kind of going with that third objective that I mentioned earlier. We want to do our best to arm these hatchery and field workers with whatever we can to ensure that they're not bringing in any pathogens from wild collected gametes and potentially stop those mortality events that we're seeing across the basin. So at this time, I would like to acknowledge the U.S. Fish and Wildlife Service, Great Lakes Fish and Wildlife Restoration Act for funding this study, the Black River Sturgeon Facility crew, and I want to give them an extra nod for this year because I went to my field site by myself and I did not do that last year, so they helped me out a lot and I couldn't have done it without them. Michigan and Wisconsin DNR, Michigan State University Aquatic Animal Health Lab members, and my major advisor, Dr. Thomas Locke, and I will be taking any questions.
Excellent presentation, Amber. Very good job. If you have a question for Amber, you can go ahead and unmute yourself or post it in the chat box.
This is Gail Curath. I hate to put my video on because it shows my office, and that's a horror. But anyway, wonderful talk, and you are very articulate. I just want to compliment on your communication style is beautiful. The information's really interesting, and of course, you can't help but love sturgeon. So my question is, I'm a little curious about the concept that the pathogens that are known have diagnostics that are difficult to, I'm not sure, I don't know much about this. What is it that makes those diagnostics like that you just can't use them as easily as possible?
So I don't have a great answer to that, but I guess what my thoughts on that would be that it's not something that we've really looked for in the Great Lakes. So when we're doing any of our routine surveillance on any juvenile lake sturgeon that are getting stocked or any fish in general, these aren't pathogens that we're really looking for in the Great Lakes season. So a lot of the people that are working in the Great Lakes don't necessarily have all the stuff on hand to be dealing with these pathogens potentially. So it's not as much as the pathogen could be hard to diagnose, more so that no one has really looked for it quite yet. So that's my best answer to that.
That's fine. One other quick question. Any plans to look at pneumovirus, since that's pretty close to you guys, right?
Yeah, so there is a subsection of my grant that will be looking at those nucleocytoplasmic large DNA viruses, but I haven't done any work with that quite yet. So
it'll be neat.
All right, Amber, there's a question here in the chat box. When you amplified the DNA polymerase from the foci, did you also see sturgeon sequences?
So that's a great question. We actually, when we were phylogenetically analyzing this, we were looking at, so I guess, yeah, so sorry. When we were running phylogenetic analyses, when we had compared it to what we were looking at, like the aspen-centered herpes virus 1 and aspen-centered herpes virus 2, we had seen a similarity of about 80 to 82%. So our interpretation of that was that we were dealing with a potentially novel aspen-centered herpes virus. And yeah, sorry, don't have a great answer, but that's...
Any other questions out there?
I have one. Excellent talk, by the way. You did a great job. Regarding the first part of your study, I just wanted to ask you, was the samples that you collected from a disease outbreak, mortality, fish kill, or was it random sampling?
So it was random sampling. I was going up to these sites whenever an agency was handling these fish and handling the adults. Additionally, if there were ever any mortalities at the rearing facilities, we would take fish for that purpose. But most of what I was doing was random sampling, just general, kind of getting a baseline for the general health of the Great Lakes lake sturgeon populations.
Yeah, so, I was going to ask if you know if there is like a mortality rate or something that you can tell.
Yeah, so we weren't particularly looking at any fish kill or any mortality events. Yeah. We were getting a good idea of what was out there in the basin.
Have you sampled any other fish species? I know that your main focus is sturgeon, but have you sampled any other species?
No, not for this project, no.
Okay, thank you.
Yep.
Good job.
Thank you.
So, Amber, I'm just going to jump in real quick on the question regarding the sturgeon sequence, if we detected any sturgeon DNA or not. And I can tell you that at this point, no, we don't have any evidence that we had any non-specific amplification. We are aware that does happen, has been documented with these primers, but to this point, it seemed like they were pretty clean sequences. And I don't want to steal Amber's thunder, but she's also got some pretty exciting results via tissue culture. Are we short on time, Nick, or?
We have 2 minutes left.
Okay. Well, I would suggest if there's no further questions, Amber, if you wanted to go into that a little bit, but you do as you see best fit.
Yeah. So like I'd said, I just got back from my field site 7 days ago today. So, but I was getting samples back to the lab in a timely manner, and we actually started to see a little bit of CPE on some of the cell lines that we were employing. And as soon as I got back, I basically ran a PCR using those Hansen primers. And in addition to running any of the lesions that I had seen potential CPE in, I'd also ran a control that was just the cells that I was inoculating the supernatant onto. And the result of that was no amplification at all on just the tissue culture cell controls, but I did get amplification on the samples that I had assayed using the Hansen primers. So it's all pretty preliminary. I haven't, since all the sequencing centers on campus are closed, we haven't had a chance to submit anything, figure that out, but just a finding that I had uncovered in the last couple days, but wasn't quite polished enough to toss in here.
Thanks, Amber, for sorry, go ahead, Gail.
Sorry, can you remind me whether or not you would expect cell virology? Do the herpes viruses that you're talking about amplify in the cell lines you used? Do you expect that?
Yeah, so I've definitely read papers that have documented like CPE happening on sturgeon-specific cell lines, but herpes viruses as a whole have been notoriously difficult to culture, so we didn't have any luck actually in my first year of sampling getting anything off of our cell cultures. So this year we modified our protocols a little bit and seem to be having a lot better luck. So were we expecting it? Kind of, yes, but we also weren't surprised when we didn't see anything in year one. So now that we're seeing something in year two, it's going to show that our modifications worked and we're now working on a different playing field.
Thanks.
Yep.
Awesome. Well, thanks again, Amber, very much for your presentation. Go ahead and stop